to paralysis and death, or alternatively, the same botulinum toxin administered in tiny quantities results in a wrinkle-free forehead. This is all biochemistry. Detailed description of these topics could easily have made it into this book, and some listeners may feel I was remiss in neglecting them along with other topics as fundamental as vitamins, hormones, chromosomes, and numerous biochemical techniques. But this is, after all, a very short introduction, and so I had to draw the line somewhere. As a result, for much of the book I focused on some of the chemistry that occurs within cells, for therein lie the fundamental chemical processes that all life shares. Finally, the boundaries of biochemistry are ill-defined, overlapping with genetics, molecular biology, cell biology, biophysics, and biotechnology. This being so, I round off the volume with 2 chapters exploring some of the fundamental discoveries in biochemistry that are influencing these other fields and society generally. Chapter 1: The Roots of Biochemistry. In many ways, the history of biochemistry is linked to the understanding of arguably the oldest uses of biotechnology: fermentation and the production of alcoholic beverages and cheese. Cheese manufacturing probably coincides with the development of agriculture and domestication of goats and sheep some 10,000 years ago. The fresh milk left in warm conditions would have been a perfect breeding ground for microbes such as Lactococcus lactis. These bacteria convert the lactose in the milk to lactic acid, which in turn cause proteins in the milk to coagulate, forming curds. The resulting reduction in the lactose content of the curds and leftover whey would have been a great boon to adult Neolithic humans, as they undoubtedly had trouble digesting the lactose, not yet having evolved a means to break down this sugar. And of course the curds can be pressed and dried to form readily storable cheese. Around the same time, other cultures made use of yeast's ability to ferment glucose into ethanol. In the process, the yeast outcompeted harmful bacteria, so rendering the resulting beverage safer to drink than untreated water. And so beer was born. Fermentation and enzymes. Fast forward to the 19th century, and an argument was brewing between 2 titans of the scientific community over the chemical processes underpinning fermentation. Louis Pasteur, the microbiologist and chemist more famous for his work on vaccination and pasteurization, clashed with Justus Freiherr von Liebig, widely known for the condenser named after him, but more importantly as the founder of the fertilization industry and father of organic chemistry. Liebig supported the chemical theory of fermentation. He built on ideas proposed by Antoine Lavoisier that yeast accompanied and possibly triggered the onset of fermentation, but once the biochemical ball was rolling, living organisms took no further part in the process. Liebig explained this by theorizing that fermentation was a result of transfer of molecular instabilities between the components of the ferment, a process that he thought was comparable to putrefaction of animal and vegetable matter. Hence, from his viewpoint, fermentation was a purely chemical process that did not require any intervention from living organisms. In contrast, Pasteur took an experimental approach. By showing that fermentation did not take place in a broth where microorganisms had been killed by boiling, Pasteur concluded that fermentation was due to the action of viable microorganisms. In his words, alcoholic fermentation is an act correlated with the life and organization of the yeast cells, not with the death or putrefaction of the cells. This statement was clearly a dig at Liebig's theories and helped to fuel the argument between the 2 scientists, which took place via their papers and letters throughout the late 1850s.